All right. Hi, folks. Good afternoon. Thanks for joining us for our next installment of the public informational meetings for hemp. I'm Zach Brim, agroecologist and hemp specialist at University of Florida IFAS, working from the Tropical Research and Education Center in Miami-Dade County through the agronomy department. So as a note to everybody, this meeting is being recorded. And will be later posted to the UF Hemp website events page. It takes about a week or so, and any of the chat information will also be saving. We will not be posting that to the website. We're working hard to make these virtual events accessible to all, so please communicate with us if you require accommodations in order to be able to participate. As a general sense of the meeting, please conduct your participation with respect and kindness. Maybe most importantly, mute your microphone. Also, to help some folks with the bandwidth, you can go ahead and mute your video. In the case that you do receive an invitation to ask a question or to speak, don't forget that you're muted. The goal of the meeting is an open and welcoming environment, and we're going to sort of go through a couple different viewpoints as we work through our conversation, really working on presenting facts and findings from our experience. The chat's going to be open. That's primarily where you can present your questions to the speakers. And as I'm not a primary speaker today, I'll be moderating the chat window. So we'll take a bit of time for our presentations and then specifically stop along the way in order to in order to field those questions. So there are several folks here in order to introduce Dr. Lance Osborne, all doctors. Doctors Lance Osborne from the UF-IFAS Mid-Florida Research and Education Center, entomology and nematology. Dr. Alexandra Ravinthi, also entomology and nematology from the Trek Station. Dr. Kevin Corris, Plant Health, Alachua County, Dr. Wael El-Wakil, and he is Sustainable Agriculture, Sustainable Food Systems, one of those cross-cutting designations. Commercial horticulture, help me, Wael.
Fruit and vegetable production.
Fruit and vegetable horticulture. There you go. Somehow he found his way around to hemp, primarily through the pests and diseases. So we'll hear from these folks throughout the meeting. If you could just introduce yourself again, cover all those key details. I'm sure the audience will be happy to hear from you. Kevin and Wael, did you want to say anything first before we get started with Lance? Thumbs up. All right, with no further ado then, Dr. Lance Osborne, I am going to give you spotlight and then you can share your screen, I believe.
It's saying I'm disabled.
Oh dear.
Oh, you disabled me.
There you go.
Okay, let's see. Let me get this up and running. Okay, how's that?
Looks good to me. Thanks, Lance.
Okay, I appreciate the opportunity to talk today. Basically, I'm just working on hemp to help some of the faculty here that have projects on hemp. I have a little bit of involvement with Dr. Ravinthi and working on spider mite biological control or mite biological control. But really, I don't have an assignment to be working on banker plants or hemp anymore, but I'm still interested in it. We spend a lot of time growing cuttings for other people. I don't know how I managed to get myself doing that, but that's where I'm coming from. Work in greenhouse protected agriculture, mainly in ornamentals. And so a lot of the technology and the things I'm going to talk about today is information that we gleaned from working on managing ornamental pests, where the threshold for these pests is generally much lower than it would be in hemp. So I think some of the systems work very well and allow us to grow hemp that's fairly clean of pests. Okay, chemical control. As all of you know, I mean, there are limited options that they don't seem to be adding very many new compounds that are registered for use in the state of Florida. I got the latest update. I think it was from April of the materials approved by the state, and they really haven't had added very many compounds. Most of the compounds would not be ones that I would generally choose in ornamentals. They're not the most efficacious. Some of them have little to no information on phytotoxicity or efficacy. So we're kind of hamstrung with the types of materials that we can use in greenhouses. A number of publications have come out that that some of the products that are being sold are contaminated with illegal products that are being used to manage pests. So I think that biological control is something that we need to look at. I'm going to show you a few slides of a crop that's been recently grown or being grown using chemical controls. In some cases, materials that you wouldn't be able to use in industry, but this is a research facility and we're allowed to do it. We're not, even with some of the good materials, we're not getting very good control of spider mites in hemp. This is a one shot. This is what the crop ended up looking like. Those are all spider mites looking for a new place to feed because the crops is so damaged. They're just webbing and spreading throughout the greenhouse. This is a plant that came from our greenhouse where we have been growing plants for stock as well as cuttings that other faculty could use. And we've not sprayed this greenhouse in over a year. And so you can see it's relatively free. At least I don't see any pests. I don't see any damage. This is the stock plants here where we are taking cuttings. This is a wife variety. Biological control is a tool and it works very well. But growers in Florida have had problems that have limited adoption, and I'll go over some of those issues. One is economics. In some cases, growers feel that it's just too expensive to buy biological control agents, and there are some companies out there that oversell the amount that people should use to get control of various pests. But I don't think that that's nowadays, in many cases, the cost really isn't as prohibitive as it used to be, especially for spider mite biological control. So economics, but it is still an issue, especially when you've got multiple pests that you're trying to manage and use biological controls on. Availability. Sometimes you may need a biological control agent for a problem that cropped up and is causing damage by the time you notice it, trying to get in natural enemies that are going to take care of the problem, especially in the numbers that you might need. It's sometimes difficult. We had that spider mite problem I showed you a few minutes ago, and it took us a couple of weeks to get materials in to use to manage it. Sometimes you run into pesticide interactions, especially in ornamentals. It's probably not as important in other crops, especially in hemp, but it still can crop up, and certain chemicals that are relatively innocuous in some cases could cause problems with establishment or just out and out kill the natural enemies. Quality control. This has been probably our biggest problem in the industry, is trying to get good quality biological control agents delivered to us and ready to be used. In many cases, it may be that they leave the seller or the company that's producing them in good shape, but they don't arrive at the research center or in the grower's hands in any kind of shape that can be useful. We just purchased like 16,000 Persimmilus and 16,000 Californicus. Persimmilus were in great shape. We could not find a single live Californicus in the bottles, and it also we had no indication that they were even put into the bottle. And so, you're not getting what you pay for in certain cases. So we stress that growers need to figure out a way, and we have methods that people can use to test the quality of of their products as they're buying them in. Another issues is seems like everything in the world likes feeding on hemp. And not everything that might attack hemp or might attack certain ornamentals can you find or purchase a biological control agent that's going to be efficacious. Luckily in Florida, it seems like we're kind of a zoo here. We can find something that's going to eat one of the pests that's causing us problems. It's just that we can't buy them in the number and the quantities that we would like to. So, and they may not even be commercially available. So one of the solutions for this particular issue, we think is what we call a modified banker plant system or an open rearing systems, where you can either grow your own, or you are basically figuring out how you can get these natural enemies into the system without really having some of the issues I just spoke about. And I'll go into these in a second. In some cases, you may not want to purchase these things or they don't sell them. So the banker plant system will fit that bill. So as an example, this heart-shaped leaf, let's say, is a banker plant. This is for whiteflies, it might be by papaya. For spider mites, it would be corn. And these are the pests that attack the corn. So this is a Banks grass mite, okay? Or if it's papaya, it would be the papaya whitefly. We can buy some natural enemies, or we have colonies of natural enemies that we could put on these plants, on the banker plant, that will feed on that non-pest whitefly or non-pest spider mite. So we use corn, Banks grass mite. Banks grass mite does not feed on hemp. The papaya whitefly will not feed on hemp. Okay, so we have hemp. This has two-spotted spider mite, or it might have broad mite, or say Bemisia. We can use those same natural enemies that will attack the bugs that are feeding on the banker plants, will also feed on the pests that we're concerned about and trying to control. Okay, does everybody understand that? So we have two different systems. We can put these plants together. Okay, so this plant has a bunch of natural enemies running around on it, feeding on these pests that don't feed on the crop. Okay, this is the crop. I'm sorry, this is the banker plant. Okay, so the squares don't go over here and cause problems, but the natural enemies will move back and forth between these two plants. So we can take these banker plants and stick them within the crop. We put them into the greenhouse or in the crop or the area where we're going to grow the crop, even before we put in the crop. So we have really a standing army, or we have these natural enemies that are running around in this greenhouse looking for something to eat. So if a pest happens to show up, we already have something there to feed on it. Okay. So we're banking the natural enemies within the system. It's absolutely critical to get this started before you even put plants into the greenhouse, because nowadays, a lot of the plant material that's moving around and we're using to start new crops can be infested by the person that sold them to you, or infested in transit. Okay. So the idea of the banker plant system is it uses alternate hosts. It uses confined hosts, such as we have a banker plant system that we're developing for mealybugs. Predatory mites are reared on alternate hosts in little sachets that contain grain. So you're using them, they're confined into an area that, in the sachets, you put them out, and the grain mites don't feed on your crop. The mealybugs, we contain them in such a way so they can't get out into the crop, but the natural enemies can fly out and attack mealybugs. There's potential to sell the entire system. I can't tell you how many growers are calling wanting to be able to buy these things. And so far, we haven't found an entrepreneur willing to try to develop and sell these, except for Carol Glenister at IPM Laboratories that's selling a banker plant system for aphids, which you can buy in. They may be specific to the region. Okay, so I can't send out papaya out of the state of Florida with the papaya whitefly on it. So it's only going to benefit our growers in Florida. It uses established natural enemies. In order to use biological control, at least one of the most important and most difficult hurdles in getting the whole system going is the establishment phase. Once you get them established, they seem to take off and you have fewer concerns or fewer problems. If you use a banker plant system, they're already established on the plant. And so when you put it into the greenhouse, it's got food, it's got a place to live. So you remove some of the hurdles. You can use it to increase natural enemies. So if you buy in a few and put them on a banker plant, natural enemies, within a week, they've increased in numbers and are healthier because they don't have to look so hard for something to eat. If you're releasing them into a crop that has that many pests in it already, you're probably doomed. You don't want to start it when you have lots of pests that would support a predator population right off the bat. We use them to evaluate the quality of natural enemies. And this was the banker plant system for spider mites proved this very early on, that when they take the predators and put them on a banker plant and nothing happens, you have to wonder what's going on. And they found that some of the natural enemies, the predatory mites that were being sold, were not laying eggs. They would run around for a few days. They look like they're a quality product, but the females were not laying eggs. And the banker plant system is the only way that we knew we had a problem. The sellers were saying that something was wrong with the plants and that they had pesticide residues. Well, the banker plants don't have pesticide residues. And when the predators didn't do their job, it was the predator's problem, okay? You can integrate it with pesticides. The plants can be removed. You can spray something else that might disrupt the biocontrol, but when you bring it in, you have the system still going. They're still established, but they're not on the treated crop. And then the predators and natural enemies move back into the crop as the residues dissipate. And all the systems I'm talking about today are safe on hemp because we've proven it. We use them ourselves. Okay. And you can grow these yourself. And I'll show you the one end of the spectrum where a commercial nursery is producing thousands and thousands of banker plants. We have systems where you can do it basically on the counter in your office or your lab. Major pests of hemps that we use banker plant systems for thrips, for mites, for whiteflies, aphids, and now we've had success with mealybug banker plant system. Okay, so mites programs have been very successful, mainly for two-spotted spider bite, but also Tetrandicus gloveri, which also gets on hemp. This is the totally different species than the two-spotted spider mite. Just an example of the damage that you see. We also use it for controlling broad mite. A very tiny mite causes a lot of damage in a lot of different plants. People across the country are saying that they're having much more of a problem controlling broad mites. We can't keep a colony going or keep broad mites alive in a greenhouse because of the natural enemies that we use. This is a commercial operation and one of the largest ornamental producers in the state. This was in Engelmann's, which was sold to Costa, and I don't think they're doing this any longer. This is corn, 2 different tiers here, and there's another 2 tiers over on this side of the room. They have them in different ages, so they rotate them weekly. They'll take the plants, clean plants. When they get to a certain point, they will inoculate them with the pest mite, Banks grass mite. And then, it's easy to tell the difference between this and 2-spot. The pattern of the coloration on the body is different. They just look totally different, so it's easy to tell. And then the grower decided he kept getting predators established in his clean colony, so he would just buy in the predators. He would grow the clean corn, then he'd put the Banks grass mite on it, and when it got to a certain point, he would inoculate predators. He would do this in his nursery, so he'd bring in clean plants that had Banks grass mite, put the predators on, and then he'd hang them up in the crop. He didn't even put emitters in here. He just waited until force the predators to reproduce, and then the plants would die and the predators would move throughout his crop. This is a stock house that he didn't spray for over a year, where he was spraying once a week for spider mites. He didn't have to spray but one time in a whole year, and that was a soap or an oil that he used. So again, he saved a significant amount of money and effort, and they had to spray these on stilts. The workers would have to suit up and wear suits and use stilts to get good coverage into these upper levels. So it worked extremely well. We were able to grow an array of different predators on this system. One system we like, oh man, this is driving me nuts here. One thing that we like is we use Feltiella, which is a small insect fly, and it flies around the greenhouse in search of spider mites, so they don't have to walk like the predatory mites do. And it's been very useful in bringing down some of the higher populations. We have Persimmilus that feeds on it. We have Californicus. Swirsky doesn't feed on it, but we use two different systems. One is the open rearing system or the sachets, or we will actually use a banker plant where we use an ornamental pepper that produces lots of pollen, and it has what we call domatia on the underside of the leaves, with the predators hide in these domatia, and they reproduce, and you can get quite a few predators and have a long-term population of this particular predator in the nursery without even having to put alternate prey in. The hemp russet mite project is in progress. We've had hemp russet mite a couple times in our at the research center here. And first encounter, we had already anticipated having mites or broad mites. We put Swirsky out. We didn't have any problems with the russet mite until we had to spray. I think we sprayed Talstar, or the crew sprayed Talstar for something, and that disrupted all of the biocontrol programs. And we think that that's when we found russet mite, after spraying Talstar, which eventually killed the predatory mites. And then we ended up with not a broad mite, but a hemp russet mite issue. Okay, very tiny. They come in often on the cuttings. We've had it imported probably twice now. We never had to, since we started using a predator, we haven't had any issues with this particular pest, and I'd say maybe 2 years now. Swirsky is one of my favorite predators, from the fact that it feeds on whiteflies, it feeds on thrips, feeds on broad mites, and there's information that it does attack hemp russet mite. We just have testimonials, and we don't have any real data to show that it feeds on hemp russet mite, partially because we couldn't keep it alive in colonies. Dr. Ravinthi would probably have much more to say about that than I do. She's working a lot on broad mites and hemp russet mite. This is the sachet that these predators come in. When we hung it in hemp, we ended up having problems with rodents. They'd come in and eat the sachets. So the sachets really didn't work that well for us after a certain point, because we had a lot of problems with mice coming in. What we did use, like I mentioned, was an ornamental pepper. This was an ornamental pepper with Swirsky on it that we put in a poinsettia crop, because they also feed on whiteflies. Okay, whitefly programs have been successful. I don't know if anybody that's listening has had whiteflies in their hemp, but a number of the growers around here have had significant problems, and a couple of them felt they lost the crop due to the Bemisia tabaci or silverleaf whitefly. Banker plant is papaya. The alternate host is papaya whitefly. And we have an array of natural enemies. We have Encarsia sophia, or transvena. We get various beetles, Delphastus being the prominent ones. And then we get, when you start mass rearing whiteflies, you start getting fungi. And we get the PFR or Ancora, or it's called cordyceps, a lot of different names for it. It builds up on the banker plants and spreads and causes significant mortality in and of itself. You can see the population of whiteflies that develop on papaya, just thousands of them. And this supports the parasitic wasp, a little tiny yellow wasp that when it lays an egg in a whitefly, the whitefly turns black, so you can tell who's been killed and who hasn't. This is an adult female. We used it as a banker plant in poinsettias, which is even more sensitive to whiteflies than hemp. And we were able to produce crops without any issues, except in this case, this is a student greenhouse. They put the papaya too close to the poinsettia. It dripped honeydew and they got sooty mold on this end of the crop. If they just put it a little further away, they would have been able to sell all the plants. We used it as for managing stock plants. This is a 11-week study. Red were the chemical controls, and the blue or the green were biological. And you can see the biological was hanging in there and doing a great job all by itself. The pesticides had to be applied at the 3, 5, and 6 weeks. And some other material was sprayed later on in the season. You can see that we did almost as well as the chemical control. If we would have hit the plants at this end with the chemical, we probably would have had no whiteflies at all. With hemp, we feel that we can develop and grow the plants at we may have a few whiteflies, but nothing that's going to be of economic importance. Now, these studies were done prior to us finding this little beetle, Delphastus pallidus, which is just a very effective predator. Once it gets in, it really takes off and will manage a whitefly population. Even if it's almost a runaway population, this predator will reproduce so fast and feed on so many whiteflies that it will clean up a crop very quickly. Larval stages, we're feeding on the late instar. They feed on adults, anything that they can catch. This beetle is kind of a nuisance for us because every time we try to run an experiment, they show up and eat everything. This is the bottom of a papaya leaf, and you can see every one of these little white things is a beetle, or the pupal stage, or a larva getting ready to molt into and become an adult. Okay, thrips. Thrips programs have been successful. We do get chili thrips in hemp, and causes some significant problems because we probably have a higher population of chili thrips than most people would, because we work on it quite a bit, but it did get into our hemp and cause significant problems. But the worst pest thrips in hemp is echinothrips. We've had a number of growers having problems trying to spray it and get rid of it. It's very difficult. It pupates on the leaf, which makes it a little easier than most, but there's no good biological control for it. Most recently, we have found that Thrips parvispinus will feed on hemp. This is the damage in a greenhouse. This was our stock house, and you can see that it caused a lot of damage to hemp. These are adults, these are immatures. There's no viable biocontrol for echinothrips except for Franklinithrips, which isn't commercially available, but we have a banker plant system and a system we use to get them into the crop. And so we are supplying people with Franklinithrips in order so they can produce their own for use in nurseries. It's a little tiny ant mimic. This is the adult. This is the immature stage. They feed on whiteflies, they feed on mites, they feed on thrips, and they have some, they will feed on alternate foodstuffs such as pollen and reproductive, the cysts from brine shrimp or sea monkeys. This is the larval stage feeding on the thrips. Here's an adult attacking a thrips. They're just extremely aggressive, and they will knock a population of the echinothrips down within days. I mean, they're just amazing. They also feed on spider mites. There's just a quick graph. I don't want to really go into it, but you can see this is the untreated, and this is the treated with the predatory thrips. The only reason we had anything going on here with mites was that one of the plants, the adults we released, we must have damaged them collecting them. And so they didn't have the inoculum and predators available early on. And then, but they did when we terminated, there were quite a few immatures. So we know that the population would have crashed. Aphids, the main pest that we deal with in our system is melon aphid. We've had a few rice root aphids. The hemp aphid came in, we had to dump everything because it was, a quarantinable pest. So we haven't had that particular problem in quite a while. Root aphids, they are there, but they really aren't causing us much grief. We use sorghum or Asclepias or milkweed to rear aphids on. An alternate host aphid would be corn leaf aphid for sorghum, oleander aphid for milkweed. We get a parasitic wasp, Ophidius, we get Diomus terminatus, and we get another little beetle called Skimnus. We can use it for aphids, green peach aphid, melon aphid, like I said, is the main issue. We know that the beetles that we work on feeds on another sugarcane aphid as well. This is sorghum with the sorghum aphid or rice or the corn leaf aphid on it. It's used in commercial vegetables as well as in ornamentals. There's a commercial, you can buy banker plants with these aphid natural enemies. Oleander, all you have to do is go outside and look at an oleander plant or a milkweed, and you will find these aphids with natural enemies already attacking them. This is a shot from my garden. We were able to manage aphids on hops, which is closely related to hemp, without any problem by putting in a few banker plants out in the greenhouse. The predators fly and attack the aphids and basically took care of the problem. We didn't have to treat for aphids on hops. It'll attract all sorts of natural enemies, from very large ladybird beetles that basically need lots and lots of aphids to reproduce and lay eggs, to the little tiny ones called Diomus that we use. That this is the leg of the aphid. This is the larval stage, removing fluids, where the adult will just feed on the whole body, just like the other one. But they don't need nearly as much food to maintain themselves. So we were able to keep a good colony on a banker plant that flies throughout the nursery and works in conjunction with the parasites. This is a shot of a parasite. Basically, they lay an egg inside the aphid. The egg hatches, and then it pupates inside, or feeds on the inside, then it pupates, and this is what emerges from the mummy. And again, we have very good success controlling aphids just by using one of the banker plant systems. And with that, I'm done.
Thank you, Lance. Really good stuff. Certainly goes over a lot of content for the pests and the ways that we have available. Couldn't be more excited about the banker plant system. That's agroecology. So nice to make that connection here with the hemp. I'm in between a couple places. Dr. Ravinthi, I'd sort of like to give you a chance to add anything, speak to your experience if you like. Also, I see you unmuting, so great. So then everybody else, we're gonna kind of split up the meeting. One side is the entomology, the pests that are invertebrate arthropods. And then the second half of the meeting is going to be pests about diseases. So I'm going to give Dr. Alex a chance to talk for a few minutes. And then if I'd like to hear from the group, have folks seen arthropod pests? Are we worried about things? Just a couple moments to chat about experiences or follow up with any questions. So Alex, I'm gonna give you the spotlight and please share with us your experience and your insight.
Thank you, Zach. Pretty much research-wise for biological control, we can confirm what Dr. Osborne showed. Amblyseius virsky appears to be the most promising predatory mite, especially for the hemp russet mite. For this invasive regulated pest, we did try to use also Neuseiulus californicus, Amblyseius andersoni, which is supposed to be a predator that feeds in this group of mites, and some other predators that we had in culture, but Amblyseius virsky was the predator that fed in most hemp russet mites within 24 hours, and it was the only predator also that this feeding was translated into eggs, so into oviposition, so that predator could feed and reproduce when feeds on hemp russet mite. Regarding control, if I have 1 minute, Zach, and I can share my screen, I can
Please go ahead. Sure.
very briefly show you the work of Maria Cannon, who does her master's on hemp. Do you see my screen?
Yes.
Okay, perfect. So let me just simply show you. We, Maria Alejandra worked with all 4 major pests of hemp that Dr. Osborne showed you: the Glover mite, the two-spotted spider mite, the broad mite, and the hemp russet mite. Pretty much, I'm not going to bother you with the details, but she created leaf arenas, and she placed the mites on the arena, and we tried these products that they are included in the hemp list that is approved by FTAX TPI. And we went with the maximum allowable label rate. So we worked with garlic oil, citric acid, sesame oil, rosemary oil, thyme oil, and mineral oil. And we did a direct and an indirect spray on this. And we worked under control conditions. And overall, I don't want to bother you with all these things. The thing that I want to show you is that the citric acid and garlic oil were the 2 best treatments in controlling the hemp russet mite, the two-spotted spider mite, again, citric acid and garlic oil as the second best, the Glover mite, and the broad mite also. In the broad mite, we also saw that let me see here, that the mineral oil was also very good in controlling it. And this is how the dead individuals look like. And what we did pretty much as a second round, we used these 2 treatments, the garlic oil and the citric acid, as treatments to generate mite-free cuttings, and using the two-spotted spider mite as our study pest, because this is the most prevalent pest that we have here in South Florida. And both treatments, both garlic oil and citric acid, were able to completely disinfest the cuttings when the cuttings were dipped in these solutions, when we, of course, compare them to water controls. So it looks like there are a couple of active ingredients, but they are also 25B exempt, that the growers can consider using in addition to biological control and other practices. It is worth stating that phytotoxicity was minimal in both active ingredients tested. That's it.
Thanks, Alex. That's hot off the press. That's exciting to see. So I had a question in my head. I wanted to follow up with the study. So you were showing the percentage of dead pests on the leaves. And what I hear you saying is those are sort of within ranges that would be a meaningful control. I was seeing maybe 80 to 90% dead. Dead mites. That was an accurate interpretation?
Yes, within 48 hours, we could already see more than half of the mite population being dead, approximately 60%. Of course, depending on the species, this could vary
Right.
between 50 and 60% within 48 hours. If we wait longer, as we did in our experiment, so until day 4 in this case, mortality in some cases could reach 100%.
Cool. So just to make sure I remember correctly and to share to say it again with the audience, it was garlic oil and citric acid.
Correct.
Okay, and one of the things that I've heard from the industry is a concern for applying oils that might find their way into a hemp extract. So the example is neem oil, where neem oil applications may be a biological, not a biological, organic control for various different pests. They're seeing signatures of the neem oil in the hemp products that are sort of undesirable. The question is sort of, would you expect similar, or would you be concerned with similar effect from the garlic oil or the citric oil? Just, curious if you found something that is a good option, wondering if some residues might be concerned.
I cannot talk to that 100% because it falls outside the area of my expertise. However, the oils are very persistent, right? They are oils,
Yeah.
they are not easily diluted with water. The garlic oil, when it is applied, you can immediately sense its presence because it smells like garlic and is very strong, the smell.
Yeah.
But in any case, when we use this type of products, we always recommend to be careful with their application, and the timing of the application plays a major role. So a potential good use of these products is maybe earlier in the production you use them, where you know that it's not going to affect the end product, right? Just to keep the population of pests manageable, and then continue with biological control until it is the time to harvest the plants.
Yeah, great answer. Thank you. And it sort of brings up the idea, these are organic, it's garlic oil and citric acid, but that doesn't mean just spray as much as you possibly can, because there might be consequences on the other side. Okay. Thank you, Doctors Osborne and Ravinthi. I would like to open up the chat window, or anybody by raise of hand if we have any questions from the audience for our entomologists. Dr. Swisher, I see you unmuting your video. Is that the intent to ask a question?
Yeah, it is. I wondered if anybody, some of these products, I'm just wondering what the cost factor is. And especially what is the cost factor compared to, more traditional kinds of practices that we've used with other crops and so on. I'm just trying to get a handle on that, how expensive it is, basically.
Alex, I'll let you answer first for the oils, and then I'll go over to Lance for the banker plants.
In general, the oils, of course, depending on the company and the product, they're a little bit more expensive. than some other conventional chemical control options. However, in hemp, we are restricted in the amount of active ingredients that are available. So I cannot talk too much about the pricing options because also the active ingredients that we have available are very restricted.
Lance, general comment on cost of Banker plant systems?
Well, no, I don't. I mean, I don't live in a real world, so I don't have to pay for this stuff. The bottom line is that it's either that or you lose your crop. There's really not. You have the options of the oils and you know they're I think they're cost effective. They're not as expensive as $400 a bottle of some of the traditional chemistry that you have to pay for. And you know, some people, you know, the oils, some of them are extremely cheap. You buy them by the 2.5 gallon jug, and you know, and they're effective. But there are side effects, like Dr. Reventi mentioned. So,
Right, and Lance, maybe one of the things you can speak to is like the cost of a sachet, or you know, so there's still a little bit of an extra upfront cost to getting your first round, or you know, a starter round of the biological controls. But the idea is once you get your system established, it's more minimal maintenance as opposed to investment costs.
one of it's something that keeps on with Swirsky. Once you have it in the system, you almost can't get rid of them. Okay, and that's why we put Banker plants to kind of feed them, because they're out there. We have to spray more pesticides at the research center for killing Swirsky, so we can run other experiments. Okay, they almost, if you're an entomologist, they become a pest. And it only costs you the price of an initial sachet, which is 25 to 50 cents, I think, something like that is what a sachet might cost you.
Thanks.
Costing them yourself.
Yeah. All right, we have a question here in the chat. Thanks, Matt. The idea that it's hot and bright, and maybe some of these oils might lead to burning or phytotoxicity. So any insight on how to preempt that issue?
Yeah, this is a valid concern. And this is always the issue when we use oil in any crop. The temperature at the time of the application is very important, because if it is above 32 degrees Celsius, which would translate around 90 Fahrenheit, then the chances of getting phytotoxicity are very high. What we recommend is very warm days to really not proceed with this type of applications. If it is a matter of saving the crop and you do want to go ahead, try to apply the oils when the temperature, the ambient temperature, is as low as possible on that day. So very early in the morning or late in the afternoon, early evening, if possible.
I agree with her. I think one of the issues with oils and some of the growers we're seeing, they're kind of novices. They don't understand some of this. The other thing is that oils can interact with other compounds, especially if there's a sulfur involved, which can cause significant phyto when it interacts with oils. So you have to be very careful with oils. But that said, they're not as bad as people think they are. There's a lot of reservation trying to get people to use oil when it's not nearly as bad as some people think it is. Just have to be careful and read the label.
Certainly. The other advantage of the oils is that they are not only effective on mites, they work very well in contact with other pest groups that Dr. Osborne showed you today. So you might also achieve control over a broader group of pests, not only one.
Including
I have
pathogens.
let me not step over that point from Lance, right? The mineral oils, the citric garlic oils may impact a biological control program as well. These are generalized sort of control measures. So Lance, I think that's what you were trying to say. Is that correct?
I'm not sure I was trying to say that, but I agree with what you said.
Oh, so then please, I'm sorry. Try again. I got excited.
No, I'm just saying that the oils have broad spectrum. They do impact natural enemies, but they also some of them work on plant pathogens as well. So.
Oh, yeah, sure. Okay. So we have a clarification question from Sam. Thanks for the question, Sam. Alex, maybe you can answer this both ways. Sort of, what was your frequency of application for your experiment? And then also, what might you expect for someone doing this practical application?
So on these experiments, we only sprayed once, and the mortality was measured based on that. And it was done in a small leaf area. We are actually in the process of working with the entire plant. Regarding how frequently someone should spray, we always recommend to sample the plant, see what you have, what pest you have, how large is the population of the pest. Then you can make an informed decision whether you should proceed with another spray application, or maybe this is the time that you should be applying biological control instead, or some other cultural practices.
I think Dr. Rementi brought up some of the most important aspects of this. I mean, what she has found out, besides treating your whole crop, but treating the cuttings and starting out clean. It's going to be an overall process. The first thing of IPM is start clean. So that gives us a tool. As long as they're registered for dipping, that's another question if that part's legal. But then it allows you to integrate the biological controls much easier than some of the classic pesticides. It's an overall systems approach.
Thanks, folks. I really appreciate the discussion and the information you've shared with us today. We're going to sort of take a pause on the discussion with arthropod pests and move into the diseases, the bacteria and fungal diseases. That's Dr. Kevin and YL. You fellas have the floor.
Right.
Well, did you want to start with your presentation?
Sure, I can go ahead and start. So let me share my screen here. You all see that right?
We see you.
All right. Well, good afternoon, everybody. My name is Wael Wakil. I am the Fruit and Vegetable Extension Agent in Hillsborough County. And for the purposes of this meeting, part of my role here is a sort of a regional support system for extension and for growers in our Southwest region in Florida, so Tampa Bay area and neighboring counties. So what I'll do here is just do a very brief summary of what we know so far about hemp foliar diseases in Florida. And before I go in, I just want to recognize all the faculty, staff, and graduate students who early on from the very beginning of this process started collecting samples and surveying plants across Florida to give us a better idea of what diseases and what problems are we seeing out in the field. And especially recognize Dr. Nick Defoe and Dr. Matthews Parrott, who provided quite a bit of the material that I'm using here today for this presentation. And Dr. Nick Defoe is actually the one who manages the hemp pathology pages on the hemp website. All the updated, and they try to keep it updated as much as possible. So I'll start with a few of the foliar diseases seen here in Florida, and just so you know, there's a lot of these pathogens or leaf spot uh, pathogens or diseases, and they might not be as problematic to growers in situations where your focus is the flower or the incidence of these diseases is relatively manageable or under control, but they can be very problematic with some of these as the incidence of disease or severity of disease increases. So one common leaf spot pathogen is Curvularia, which has been noticed in up to 70% infestation in field trials. And you can see the symptoms are kind of small yellowish spots that turn into tan, and they have a brown edge around them and a yellow halo. Another common one is Cercospora. Cercospora is a very common leaf spot pathogen in a lot of different crops. And for hemp, we see here that it starts with a small yellow flecks or little white, yellowish spots on the lesions with a yellow halo. And as the disease severity increases, it can pretty much uh, cause leaf chlorosis for the whole leaf and can cause defoliation for the whole plant, depending on the severity of the disease. Another very common, I think the most common leaf spot is Septoria. Septoria, it causes these brown spots with yellow margins, and advanced symptoms. You'll see that the whole leaf starts yellowing, and sometimes you'll see the whole plant stunting and maybe even kill the whole plant, depending on how severe the infestation is. Gray mold, again, these are some of the most common ones that we've seen. Gray mold is an opportunistic fungus that causes a lot of different fruit rots or gray molds on plants, and it's very common to see that gray mold on strawberry plants, especially. So if you've had some strawberry plants sitting in a clamshells for too long in your fridge, you'll probably see some of that gray mold that is Botrytis cinerea, and it has a very wide host range, and it can cause it's important for hemp because it affects the flower buds usually, and it can come in through natural openings or any wounds from say insect damage or any mechanical damage on the plants as well. And the symptoms can also progress to the point that they can start affecting the leaves and causing um, stems to break, and it can also cause damping off and small seedlings as well. Dansomonas, again, is one of those also very common bacterial foliar pathogen or leaf spot pathogen, and we've been able to identify two different species of the Dansomonas in North Florida and South Florida, and it starts as these black little specks or brown dark specks with chlorotic halos, and really there is limited management that you can do normally to bacterial disease in general. But one thing that you can do is to sanitize and clean your equipment or start with clean material, like Dr. Osborne had mentioned earlier. You could also, early on in the season, you could potentially remove some of the infected material to reduce the inoculum, the amount of inoculum in the field. However, that may not be as feasible if you have a big rain event or something like that that basically spreads and splashes all those bacterial cells all over your crop. Again, if you hopefully you're not overhead irrigating your hemp, but if you do, that's one thing that can help exacerbate the problem with bacterial diseases. Bipolaris leaf spot is another leaf spot pathogen that has been detected in Quincy, Florida. And you'll see this tan colored spots with dark brown margins around them. Downy mildew, again, one of the most common diseases in a lot of different crops. Pseudoperonospora is a very common genus that causes this downy mildew disease, and what it looks like, it's brown to blackish spots on the lower surface of the leaf, and on the upper surface of the leaf you can see this yellow necrotic spots. And if the environment is ideal and there's enough moisture and leaf wetness for a prolonged period of time, you can see on the lower surface of the leaf, you'll see this gray fuzzy mycelial growth that is purplish sometimes in color. And I know this is a foliar disease presentation, but I just wanted to, the root knot nematode, this is an honorable mention. This is one of the most underestimated plant pathogens or plant problems is Mildigine incognito or Mildigine species. This is a very, it's a nematode that has a very wide host range. And usually it's a problem if you're planting in an open field. Hopefully it's not a problem if you're able to fumigate your fields or if you fumigate your beds before planting. That won't be a problem. If you're growing in a greenhouse, you're likely going to be using sterile soil, so that's not going to be a problem for you. A couple other pathogens that have been seen in other states that we should be on the lookout for is powdery mildew. Again, a very common pathogen across different crops, and you'll see this powdery growth on the top of the top of the leaves. And usually, it's a problem in greenhouse environments when it's very humid and very warm. That will increase the intensity of the disease. So anything that you can do to reduce the moisture and aerate your environment in the greenhouse. So if you can space out your plants or move a little bit of that moisture with fans, that could help reduce the favorable environment for that disease. Rust is another common disease that affects a variety of different crops. This particular rust causes, you'll see this on the left, on the picture on the left there, you'll see on the front and the back side of the leaf, orange and brown rust colored lesions. And on the back, you'll see these pustules that basically erupts and releases thousands and thousands of these spores. And for most rust pathogens, they usually have an alternate host, which means it needs another host to complete its life cycle. It's not completely understood the life cycle of this specific species, or at least to me, I'm not too familiar with this specific species, but this is also something to keep an eye on. And as general kind of leaf spot disease management, where Lance has mentioned this, we're very limited to what's available out there for fungicides or bactericides. Most of what's out there is biological control agents or biorationals, which again, I completely agree with Dr. Osborne, would not be my first choice of control, but unfortunately we're limited to some of these. So anything that you can do to avoid having the disease, or any cultural practices that you can do to reduce the problems that before starting, that would be great. So controlling things like weeds or secondary hosts, or cleaning your fields or in greenhouses where there's no residue or any plant material that any of these pathogens can survive on or overwinter on, that would be great. Again, avoid things that can increase your leaf wetness. So overhead irrigation, definitely try to avoid that. Improve aeration in greenhouse areas. And a lot of these pathogens, they all kind of look together, look the same, all these leaf spot pathogens. So if you try to, if you need help with identifying some of these, we have plant disease clinics all over the state, with the main clinic in Gainesville. And this is just a map that shows approximate locations and some of the addresses there if you need them. And this is just some overall sampling best management practices. If you're having a leaf spot problem, please send enough of the plant material or the plant shoots to the clinic to be able to identify what it is. If it's a wilt disease, send the whole plant if possible, and hopefully plant that has not completely died and mummified, because the diagnostician is not going to be able to do anything with that. If you have bigger field plants or fiber plants, for example, that may not be feasible to pack in a box and send it to the clinic, take some pictures and give them a phone call and see what they're asking to do in terms of material that you can send to identify the problem. With that, that's all that I have, and I put a QR code to the plant pathology or plant diseases page on the hemp website. So feel free to visit that for up-to-date information. And take any questions if there are any.
Thanks, Dr. Awakil. I don't see any questions in the chat currently, so we'll go on to Dr. Corus and follow up if there are any. Kevin, I see what amounts to a
So we go.
your window not yet to slideshow mode.
Let's see. There we go. Are you seeing presenter mode or the mode you're supposed to
No,
see?
that's good. You ready to
Cool.
go?
All right. Okay. So thanks, Weil, for kind of covering the top of the plant, or the leaves anyway. And so now I'm going to go ahead and kind of talk about the diseases that we've seen here in the state affecting the roots and crown and lower stems of the plant. And unfortunately, these are the ones that we can that can cause quite a bit more of an issue when trying to manage them. But stepping back, my name is Kevin Corus, and I am the Ag, Row Crop, and Natural Resources Extension Agent for up here in Alachua County. Doctor of Plant Health graduate from the University of Nebraska, and kind of a background in plant pathology and plant disease diagnostics. But anyway, so we're just going to go through some of the things that we've seen, and I won't be a broken record here, but just like with the insects and the foliar plant pathogens, our control options are limited. So we'll talk about some of the cultural and mechanical and types of options that are still available, and show you where you can go and reference the latest available products. But again, not a lot of inorganic pesticides. None so far are registered for the control of plant pathogens in hemp, but again, broken record syndrome there. So moving on. The first thing that we have noticed are seed saprophytes. So what is a saprophyte? Basically, it is a microorganism, typically a fungus or a bacterium, that's living on the outside of the seed, but it's a microorganism that's always going to be associated with that seed. When we try to take that seed and grow from it, we put it on growth media and try to get a fungus or a bacterium to grow out from it. We'll often find these. And so we're seeing some consistent organisms show up with that. But so far, none of them have proven to be pathogenic or inhibiting seed germination in any manner there. So that's good. So Rhizoctonia, damping off, among other organisms that are causing damping off, big issue. It can be difficult to diagnose what's causing damping off, and we'll kind of go into that, and I'll show you some reasons why it's difficult to know exactly which pathogen is causing your seedlings to wilt and die just by looking at the seedling. And I'm really glad that Weil put up that graphic of all the different diagnostic labs across Florida, because you really got to utilize that. And you can't start the right corrective management strategy unless you have the right diagnosis made. So you want to make sure you're after the right pathogen if that's indeed what's causing any kind of plant collapse. So Rhizoctonia, or Fusarium crown rot, another one that we see here, Rhizoctonia not only causing damping off of the seedlings, but causing a root rot in mature plants. Southern blight, hemp canker, and stem canker are the ones that we'll go through. And then after each name there, you can see kind of where we saw them. So at either a research site or in field at some of these areas. So Gainesville, the Tropical Research and Extension Center, North Florida Research and Extension Center, which is the Quincy location, and those other other locations. MREC is Mid Florida Research and Education Center, for those that don't know. So nothing surprising here. These are the same diseases that we've seen show up in other states that are growing hemp or have been growing hemp for a lot longer than we have, i.e. Kentucky and North Carolina. This, the list that both Weil and I are giving to you here is in no way inclusive of all the plant pathogens that can infect hemp. In fact, I think I just looked, there are about 45 recognized through the American Phytopathological Society. We're not going to see them all here, but I think we have the potential to. But so we'll focus, though, for the sake of brevity, on the ones that we have seen here. So again, going back then, we talked about seeing these recurring seed saprophytes. So rhizopus is another pretty common fungus. That's again another like bread mold fungus you'll see in your fridge if you leave it in there too long. Alternaria is ubiquitous. It's found all throughout nature. You're going to find Alternaria anywhere you look, to be honest, out in nature. If you take a sample and prepare a slide, you're probably going to find Alternaria fungi in there. So not a big surprise that they're associated with hemp seeds. Cladosporium and Fusarium as well. And again, Fusarium, finding it associated with a seed is not uncommon. Many seed saprophytes are within the genus of Fusarium. And then several different bacteria. So you can see there's some growth media plates there. The one on top there, you can see all that fuzziness with the black dots. That's just I'm not even sure which, that's Rhizopus there. Yeah. That's fruiting, clearly putting out its spores. And then you just see some other both fungal and bacterial growth in that bottom picture that comes out when we plate these seeds onto growth media. So again, nothing, jaw dropping or eye opening there. Just kind of expecting to see all this. And again, we'll keeping an eye on some of these Fusarium species to make sure that they're not indeed pathogens or seed germination inhibitors in any way. Because again, we see damping off, and we know that there are several different species of fungi that cause damping off. So damping off is basically just referring to your seedlings come up, it germinates, and then it just wilts and dies. Or maybe it doesn't even come up, doesn't break the surface. And when you dig that seed up from the soil, you see that maybe it had germinated, but the hypocotyl got infected right away, or the radicle got infected right away, and that it just didn't even break through the surface. So you can kind of see some of those bits and pieces there in that photo. So one of those was Rhizoctonia. One of those is Pythium. Can you tell from just looking at the symptoms? Well, not really. Maybe a super trained eye could tell. Rhizoctonia typically infects at the soil line. So you'll be able to see a lesion develop right at that soil line on the hypocotyl. Pythium typically infects through the root apex, right? So it's coming up through the very tip of that root, and it's working its way up from the bottom to the top. So maybe some symptoms could be noticeable, but really what you need to do is so that you can target your management plan is to send something like that into a clinic so that they can use the tools available to them to figure out what exactly is causing the problem. And most of the time, simple microscopy can be used to do that. And that's what we're seeing here. Is on the left there is a photo of some mycelium, or some hyphae rather, of the fungus Rhizoctonia. And I think you can see my mouse when I move it, I believe. And so Rhizoctonia is identified by these 90-degree branching hyphae that have septa right after the branches. And so there's actually typically another septum down here. And so we call this the peace pipe branch. So if you were to take just this part where the septum is here, here, and there is another one down here. And you kind of turn that on its side, it looks a bit like a peace pipe. So
Sorry,
that's,
Kevin, I'm going to interrupt you. We
Yep.
do not see your mouse.
we do not see the
I don't
mouse.
know
so
if you're
that's gonna make
operating
it
on
hard.
two screens.
Oh, well,
There might be
no.
a
Well, wait, how
control
about
on the,
now?
that's it. Yep.
Oh, you see it now? Okay, sorry. So this is what I was referring to. So Rhizoctonia is the hyphae that we're seeing growing here on the left-hand side. And you see this perfect 90-degree angle of a branch. And then right after the branch, there's the septum. Up here, there's a septum. And then down here, there should be a septum, which is basically just a cell wall. And if you were to cut this piece out from that septum to this one up here to this side branch septum here, that little piece, if laid on its side, would look like a peace pipe. Put the tobacco here, etc. So we look for those kinds of tiny microscopic little details when trying to figure out what exactly the pathogen is that's causing this. And so if we look at that picture on the right, we see these spheres, those little spherical looking, globular looking little pieces of material down there. And those are Pythium oospores that are embedded inside of this plant. So what you're actually seeing here is plant material. All of these long rectangles is an individual plant cell. And so anyway, we were able to, a root cell. So take a very, very, very thin section of that root, put it on a microscope slide and be able to visualize the spores of Pythium. Then we know exactly that what we're dealing with, and we can kind of tailor make our management according to that. So there's not a lot of differences what you would do between Pythium and Rhizoctonia or Pythium and Fusarium. It is good to know the difference though, because Pythium or Phytophthora water molds have a little bit different biology. They're a little bit different growth habits, temperature regimes, and things like that. So it is very important to figure out what exact plant pathogen you are dealing with. And again, you may need a plant disease clinic in order to do that. So Fusarium can also cause damping off. And so basically, if infection occurs at that seedling stage and it dies, it's damping off. If the seedling is able to survive, but is still infected, that infection will often turn into a crown rot or a root rot later on, or a stem rot later on in the season. So even if it doesn't escape early season infection and damping off, unfortunately, that pathogen can kind of come back to wreak havoc a little bit later on in the season with a stalk rot of some kind. And Fusarium is definitely one that's known to do that, not just in hemp, but that's a common issue in other things like corn. Early season infection leads to stalk rot later on in the season. Rhizoctonia root rot is again another thing that we have noticed in our protected ag plants in our potted agriculture. For whatever reason, pruning appeared to help a little bit. And there were a couple of varieties that did show some resistance. I don't know that they were selected and looked at for developing for further resistance or trying to find a good mother plant to help breed into some of our stock. I can't speak to whether or not that was something that was done, but it's promising that there was some varieties that did show resistance, which hopefully there was the same type of inoculum load to all those plants and it was true resistance that they were noticing. But again, we really haven't been able to look at this long enough to develop these kinds of studies to confidently say and advertise that we have seed stock out there that contains good, true resistance to these pathogens. It would just take a lot more breeding and research efforts to say that confidently. Sclerotium rolfsii is a fungus that causes southern blight. This is a pretty common one. There's a couple of fungal stem rots that have similar symptoms with this white fuzz that kind of gets produced on the lower stem, kind of towards the soil line and working its way up a little bit more. What you will see in this bottom left-hand picture is very typical of Sclerotium rolfsii, where you see these little black dots. Each black dot is actually connected to a tiny little thread. Again, this is the fruiting body of this particular fungus. I actually believe that those black dots are sclerotia. And they're basically hardened masses of mycelia that can survive very readily in the soil. And so these sclerotia are some of the biggest pains for us when trying to manage these particular stem blights, because these hardened masses can stay viable in the soil for a really long time, and their biology, the way they're put together, these sclerotia, these blackened hardened masses, helps them to resist dehydration, water loss, and degradation from UV, because they're melanized and suberized at the same time. So they can hold water within themselves if they need. They don't lose it to the environment, and they can protect themselves against UV. So these sclerotia are really nasty things to try to control when managing these diseases. And another issue too with a lot of these stem blights, especially this one and the next couple we're going to talk about, is that their host range is rather large and includes many of the important and common agronomic crops that we grow here in Florida. So one of the the best management recommendations for pathogens that survive readily in our soil is to rotate to a non-host crop. Well, that becomes very hard to find when you have a pathogen that has such a large host range. So again, this is very, very similar to the last disease, except hemp canker doesn't necessarily form on the soil line. It can form a little bit higher up on the stem. It can also form right at that soil line too. But what you're going to notice again is that white cottony growth. The pathogen for this particular disease, Sclerotinia sclerotiorum, also produces sclerotia, as you can hear in its name. And the little arrow there is pointing to the sclerotia. Again, these little black hardened masses that can survive in the soil. And then when temperature is right and when there's a host nearby, they can sense that, and then they can basically germinate from that hardened black mass and create infection pegs and create a disease cycle all over again. Even after several years of a host not being around in that soil, those little black hardened masses, those sclerotia, can unfortunately germinate and cause disease. So just again, management considerations for these kinds of things. There isn't a whole lot we can do. If you're able to sterilize the soil, if you're out in the field growing and you've got 2,000 acres, it's not something that's reasonable. But if you've got a small plot, you could try soil sterilization. If you can get it hot enough, down deep enough, you might be able to get some control that way. If you're growing in a greenhouse or protected agriculture, you could steam sterilize your soil to get rid of these sclerotia. A little bit more manageable in protected ag, harder to manage out in the field situation. And again, crop rotation is complicated by the fact that these particular fungal plant pathogens have a broad host range, and a lot of those hosts are agronomic crops that we plant. Another one is stem canker, Diaporthe phaseolorum. It, as the name implies, creates really large, nasty cankers on the stem, which will eventually cause either entire branches or the entire central growing stem to die back. Basically, the canker is a true canker. It blocks the water conducting elements, and the plant collapses. You can see what it looks like there through various phases of infection, kind of getting progressively worse as you move to the right. And then you can see the fruiting bodies produced. These little black dots on picture C will give way to the conidia that you are seeing, the spores in picture E there. And it has both alpha and beta conidia. You can see one's kind of a longer fusiformish, and the other one's a small little football looking spore. I believe this is another fungus that's capable of producing sclerotia or those hardened masses. I believe that's what we're seeing in this media dish here too. So again, very difficult to control once it is resident in your soil. And broken record, but there are no chemical options available, commercially available for use against plant pathogens in hemp. So cultural management, of course, is critical. So if you can avoid diseases, rotation is key, but you gotta understand which pathogen you're trying to rotate away from. And if it's something that, again, has a very large host range, you don't want to put something in the ground that it's going to be a host of as well. So make sure you're understanding which pathogen you have and you're putting something in there that's not a host. Plant spacing can actually help. This is especially more important for managing the foliar diseases, as Wael was saying. Anything to encourage sunlight and airflow in the canopy will help reduce leaf wetness, which will indirectly help reduce the amount of foliar fungal blights that can develop. Sanitation. So remove diseased plants, diseased plant parts, remove from the environment. Don't just stock them on a pile next to the field. Remove, burn, or bury them if you can. If there are weeds nearby that may also be hosts of these particular pathogens, you want to make sure you're controlling those weeds as well so they're not harboring inoculum. Biologicals and organics, again, there's not a lot out there, even less so for diseases than there are for insects. I don't have a single one, to be honest, to recommend in terms of biological. I don't think we have anything developed at this point. And then in terms of organics, you can get more information on pesticide availability through this website here, but I would actually rather point you to the Florida Department of Ag's updated list that's available at this website. Again, most of these are inorganic. All of these are. There isn't a single organic or fungicide that's labeled for control of diseases in hemp. So I would recommend looking at this website and checking it often to see if anything's been added to the list. But again, most of these are preventatives, trying to keep the fungus from getting in the first place. And most of these are going to be developed to help protect against the foliar blights. None of these products are going to give you really good control against any of the soil-borne root and stem and crown infecting pathogens that I've talked about. So wish there was a better picture to paint with that. I think our best foot forward in terms of dealing with plant disease is putting more effort into breeding away from susceptibility and towards resistance in our lines. And with there, hopefully be able to manage most of the problems that we get. Again, don't forget to, if you have a problem that you're dealing with or seeing out there and you're not sure what it is, consider sending into one of our disease diagnostic clinics. You can't start the right management strategy until you have the right diagnosis. So with that, I am done with my segment too. And we did want to leave a little bit of room here at the end for some discussion or questions on either insects or disease. And yeah, with that, I can hand it back over to you, Zach, and thank you much.
Sure. Thanks, everybody. Please don't run away. We got a poll here for you, so I'm going to just give a couple of minutes to open questions for our disease team, and then I'll open the poll to wrap up the meeting while we continue our discussion. So from the chat or wave of hand, is there anybody that has questions for Kevin or YL about what they were talking about with the diseases? Okay, the repetition was helpful, Kevin. It seems like everybody got it. One of the things I want to add about the disease, pest and disease clinics, very much an important resource for IFAS and our producers. Just to mention, hemp is a little bit of a special case, as everybody's aware. So if you are sending in plant material to a clinic, then you'll also want to send in the permit, or a copy of the permit, more specifically, that acknowledges that you have the legal authority to possess and distribute that material. John in the chat is just saying it's good information and thank you. So I'm gonna count to 10 and see if anybody else wants to add something. Okay, so I've gone ahead and launched the poll. It's just 2 questions. And so please go ahead over there and push those buttons on your way out. Also, I wasn't able to find a way to do an open-ended question in the poll to do a survey. So I just dropped a couple questions in the chat. So you're still welcome to put in a question to any of our speakers for today. But also just about halfway through the year, looking to catch up on some of your feedback for our group. So in the chat, if you'd like to let us know the most important thing that you've learned from IFAS. Also, let us know if there's more information that you need, sort of topics that you have heard or haven't heard. And we'll be sure to get to you. Our last meeting of the year, we're leaving open to hot topics. I'm going to try as much as I possibly can to avoid having to talk about Delta-8 THC in our last meeting for the year. So far, that's winning the hot topics anyways. I see some folks completing the poll. Thank you. And I don't see any other comments in the chat. Any of our speakers want to add anything to wrap up our discussion today? I was
Thanks
really
for having
hoping.
us.
Oh, go ahead, Lance. Sure, you're welcome. Thank you. I was hoping to hear from a couple farmers today. I know it's a bit of a challenging situation to acknowledge that you've been a victim, if you will, of a pest or disease outbreak. We all experience these sorts of things. I have a graduate student waiting in the wing to share his experience, lacking others. But anyone from the group have an issue that you've seen? Hearing about something of major concern? How about what are sort of some of the things you've experienced in related to pests and diseases? Vicky, you want to share with us something?
I do. I think that another way of looking at this is through the lens of ecology, ecological farming. And I think for a lot of our undergraduate students, if you could put this together in a way that talked about the ecology of farming and eco-farming, ecological farming, I think you could really capture the excitement of some of our undergraduate students.
Okay, interesting. And we kind of started off that discussion with one of those examples. The Banker plant system is very much an ecological way to develop our systems beyond just pest management as a pest management tool. So certainly some of the information that Kevin shared about how to rotate your crops or sorts of the biological expectation from some of these diseases. All right, Mickey, we got some work to do. Thank you, Dr. Swisher, social science expert, helping us build some practitioner communities. I see Ajit has unmuted his video, so he helps me operate. He does all the work of organizing our hemp nursery, a greenhouse operation. Boy, over the last couple of years, have we experienced some pest and disease issues. So just with a couple of minutes left, I invited Ajit to kind of share his experience. If nothing else, to help some folks kind of understand what you might be up against, or if you've had this sort of experience, that you're not alone. So IFAS is here to help, and maybe you can sort of give us an idea of what we've seen, but also maybe more importantly, what we've done to ID and to address some of these issues. Go ahead, Ajit, you can take a turn.
Thank you very much for the introduction, Zach. So yeah, just about I want to say about 75% of the pest and disease that I have seen, everyone mentioned today, we have actually experienced in the greenhouse. So all those mites, aphids, whiteflies, some of the fungal disease names I'm not too well at saying so, but many of those we've experienced down here. And I am very appreciative to hear the need or the interest in ecological, that ecological approach, because I think at least with the temperatures and the conditions here in South Florida, specifically Homestead, it can be very extreme. So, we've had, especially for instance, with powdery mildew, within 3 to 4 days, it's very easy to see powdery mildew come back after a spray. With certain mites, such as the spider mites, it is a constant. If we are not spraying twice a week or every other week, there is a chance for us to start seeing issues. So it's definitely something where an IPM schedule is necessary, along with, and I really appreciate Dr. Lance for saying this, but along with a clean system, a clean plant at the beginning is going to have that much more of an advantage of overcoming certain attacks from a pest or a disease. So that's going to be very important in moving forward for many of us who want to continue this hemp industry. But I am happy for everyone to continue working together and just, identifying some of these pests and disease and how we can utilize the diagnostic centers for building this database on what's out there. What is out there to attack hemp and how can we, mitigate those risks and manage those type of issues.
Thank you, Ajit. Maybe just to clarify, you mentioned that we have been doing some sprays. If you can be more specific about what that is and why we've chosen to do that.
Yeah, so I have been hesitant to go on to a full program for the past 2 years. So granted that we've mainly been using Sulfoil X, which is a general petroleum oil, as well as Avid, which the active ingredient is but yeah, Avid is the spray that we'll use for our typical soft body insects. So with the mites and the aphids, sugarcane aphids specifically, we've been using that to control them anytime we see their population in the greenhouse. The Sulfoil X we've been using on a weekly basis, and that's again mainly for the powdery mildew and larger insects. So it is a deterrent, more of a preventative measure. We have also used Bioseries WP, which is a soil drench, and that has actually helped with the root aphids. But again, it's something where if you're not doing this on a bi-weekly, monthly basis, for the conditions here in South Florida, they can get out of hand fairly quick. We've recently obtained some BioSafe system products, a whole line that I am excited to try because I have pretty good experience with the Bioseries WP so far. So, and yeah, while I was in the industry, had experience with their products as well. So I have found that with proper spraying, it can do magic. Keeping to the schedule is a key. I'll leave it at that. If you have a schedule and you stick to it, you will see better results. But unfortunately, with hemp being new, sometimes identifying what these symptoms are and knowing the root of the problem also takes time.
Thanks, Ajit. Just a couple points of clarification. Perhaps to the point of the schedule, our operation includes a monitoring step along with that schedule, which is perhaps the critical step, making sure that we're out there regularly looking for things. Also, I want to be very clear that the regular sprays that we've been conducting has been the horticultural oils. And so those are sort of available to commercial hemp farmers. The chemistry that Ajit mentioned is something we have used in a rare instance as needed to maintain our genetics collection. But for using that chemical currently off label, we would have lost the material in our greenhouses. So these plants are not to go into commercial stream. They're under research program. So that chemical would not be available for a commercial operation. And so I just want to make sure that that's clear. I'm going to end the poll here just so I don't give the answer away to everybody, but just to say that the number of inorganic fungicides or pesticides registered for use on hemp in Florida remains at 0. So most folks got that message, but just a point of clarification. So a couple questions thank you, Dr. Osborne, for joining us today. I'd be happy to share, or get us in touch if anybody has any follow-up questions there. There are a few questions on the chat that we can work through. So what do we do with the plants after they've fully matured? And sort of after the testing, once the crop is matured, what happens to the crop and the plants? For our research programs with these sort of experimental systems that we're putting up, our plants go to destruction. That's one of the agreements we've made with FDACS related to how to operate our research program. In the commercial side of things, folks prior to harvest would have to submit plant material for testing. That is a certificate of analysis for THC, Delta 9 THC, less than 0.3%. With that compliance verification, then a producer can go along with the material into harvest and sales. The COA related to approval of harvest is limited to that Delta 9 THC designation. However, one thing that I would like to stress as part of today's discussion is that what folks are considering a full certificate of analysis that includes the cannabinoids, the terpenes, and for the purposes of our discussion today, also the heavy metals testing, trace metals, and the organic contaminants, the pesticide residues. That's a really critical piece here for folks to be considering as we think about crop quality moving into the stream of commerce. Has anyone had any luck controlling caterpillars or grasshoppers? And perhaps also a clarification, does anyone know if there's a BT product that's been approved on the hemp list? Can one of our other speakers answer that question?
Nothing comes to mind. I haven't seen the most updated list, but nothing comes to mind right now about
Okay.
BT.
Kevin, do you remember any products that have BT labeled?
I don't.
Okay.
Again, it would be, let's see, I think it would be listed in that
Right on that
the
screenshot
FDACS
that you showed us.
website. Yes, sir.
Sam, we'll have to get back to you with an answer there. I think you're thoughtful to point out that the caterpillars and the grasshoppers have been an issue. I've seen the grasshoppers and the locusts. I haven't seen them take out my crop, but I've certainly heard from others where folks have lost crop to the grasshoppers, the locust. The caterpillars is definitely something that we've seen. Okay. Thank you, Nicole. Dr. Casuso from FDACS has shared with us the list in the chat. and she's acknowledging that there are a couple listed with the BT. So you're not without a tool in the belt there. So Crimax and Dipel. Okay. Thank you, Alex. Any other questions? Parting thoughts? I'll leave the discussion open to the group, to anybody participating. Okay, well, with that, I want to once again thank our speakers, Doctors Lance Osborne, Alex Ravinthi, Wael Ilwakil, and Kevin Corus. Thank you, everybody, for your participating. I see some clappy emoji, and thank you, everybody. I think this is a lot of critical information. Again, we'll be posting the video and sharing some of this information. I'd also like to acknowledge that there is a hemp pest identification guide that is available at the IFAS extension bookstore. So we do have a material that's available. I'm just, pulling out the example here. So we have this very handy guide here for the arthropod pests. We're also very close to developing something similar with the diseases. So a lot of this information has been coming along for the last few years as we give this a try in Florida. And so this is just the surface of the information that we have available at IFAS on pests and diseases. The pest team has a website there for the insects and the invertebrates. The disease team has a similar website as well. If you have any questions, I'm sure there's a way for you to find us, and you can certainly reach out for more information. And just a couple more deep breaths before I release everybody. All right, everybody. Thank you very much. Have a good day.